Welcome to Molecular Forecaster tutorial on CYP metabolism prediction. In this video, you will learn how to predict CYP-related properties using this module, and we will look at the metabolism of imipramine as example. To begin, you can select your working directory on clicking on Browse, and then we are ready to start forecasting. The first thing that we need to do is to import the molecule of interest. This can be done, for example, by downloading the SDF structure from, for example, PubChem here, or you can use the sketcher to build your molecule within the program. To do this, you can click on the cog, which will open a new window, and now we can start drawing our molecule using the fragments on the panel on top. Let's start with the 7-member ring, then add the benzene rings. Now we can draw the alkyl chain. And finally add the nitrogens by putting the mouse on top of the position we want to change and then click on N on the keyboard. And we're done. Now we can click on File, Save Structure as SDF. Since I already have the file, I will click on Cancel, but you should click on Save. Next, we will go through each section of the workflow and review the parameters so that these exclamation marks disappear. For this, we can click on the cog and then review that everything's in place. For example, that the input file is the correct SDF. When you're satisfied with the parameters, you can click on Save and Exit. Notice that now we have a little green check mark at the bottom of the module here, meaning that everything's been taken care of, and now we can proceed to the next part. If you click on the cog again, review that the parameters are correct, and click on Save and Exit. And finally, for impacts, here I want to make sure that I select Site of Metabolism Prediction with the little check mark. And for the P450 enzymes, you have a selection of 7 different ones. You can also choose to run on all 7 or upload another enzyme of your choice. But I will select 2C19 for our case. And then again, click on Save and Exit. And now we are ready to run the workflow. When the calculation starts, a new window will pop up. This is the log of the process that you can monitor how your calculation is going. Since I am on a Mac, this is what it looks like for me. If you're on the Windows, the terminal will be black, and on Linux, a new shell will pop up. When the calculation is done, you will see some sort of success message. I have process completed, but it might slightly change depending on the operating system. To see the results, you can click on the magnifying glass in the last step of the process. And here, before proceeding, make sure that you select again the P450 enzyme with which you have run the calculation. For us, it's 2C19. And here you have 3 options for the results. You can see the metabolites in the binding site, in the full protein, or as 2D. So let's start with the 2-dimensional structures first. So here we see the top 2 metabolites of imipramine, and let's compare them with desipramine, which is a known metabolite. As you can see that here the prediction is the oxidation on this methyl, which is the first step for demethylation, leading to desipramine, as we can see. Therefore, here we're on the right path. One thing to keep in mind, though, is that the scores assigned to the metabolites do not correlate to the reaction rates. Either of the top 2 structures are predicted to be the major metabolite, while the rest are less likely. To see how our molecule fits within the protein, we can see the metabolites in 3D. In this window, you can see the full protein, the heme center, and both of our metabolites. You can unselect one of them here in the data table below the visualization screen. Now if you zoom in, you can inspect carefully how your molecule is actually placed within the binding pocket and investigate whether some interactions with other amino acids in the binding pocket can be used in order to modify your molecule and disrupt the metabolism. apart from blocking the site of oxidation. The details may be a bit difficult to see, but you can also use the metabolites in the binding site to have a more zoomed in version. This wraps up our tutorial on CYP's metabolism prediction. We hope that it was useful, and we'll see you next time.